Good morning, everyone. Thank you for coming. We always schedule the best talks for first thing in the morning. So our first session is Trace, Fire, and Pattern. And our first speaker is Dr. Kelly Meiklejohn, Associate Professor at North Carolina State University.
Thank you, Greg. Okay, can everyone hear me okay? Okay, perfect. And I've also got Dr. Jack Hippis here today, who's also going to be presenting alongside of me. So we're going to jump right in. Our project that we had funded by NIJ that started in 2022 is on the assessment of the added value of new quantitative methodologies for the analysis of surface soils in forensic soil comparisons. So the standard disclaimer here. So today we're going to be talking about forensic geology, and soil and dust can be submitted to a forensic lab associated with a criminal investigation, and it can be very useful in that investigation because soil is spatially variable. It can be inadvertently transferred to objects and people, and so it can potentially be overlooked by a perpetrator of the crime. When this evidence comes into a forensic lab, an examiner is asked one of three typical questions. The first is identifying whether that is geologic material. The second is determining that question to known comparison. Does the soil on the shoe match that of where the victim's body was buried? And then the third question can be, where did that soil come from? Is it from North Carolina or is it from Oregon? And so to do this, the forensic examiner will typically look at the inorganic components of that soil material. And so those are things such as colour, the minerals that are present in what quantities, and they use a range of differing expert methods to achieve that goal, to characterise the inorganic components. Alongside the inorganic components, there's also the organic components of a soil sample, and this includes things like pollen spores, diatoms, insect and plant fragments. And while a forensic examiner can recognise those, typically to identify those to a specific species requires specialised expertise and morphology, so it's typically outside the wheelhouse of a forensic geology examiner. So when specifically looking at comparison of soil, so that second question that gets asked when geologic evidence comes in, there's sort of three main outcomes for analysis. So the first is there is a possibility that the questioned soil is originated from the same source as the known. There is not a possibility that the question originates as the same source as the known, and no conclusion. And this terminology is taken from the OSAC standard for forensic geology, one of their standards. In order to make any of those conclusions, this relies on having sufficient inorganic material for the examiner to analyse, and also exclusionary differences. And so there are some situations where there may not be sufficient inorganic material or exclusionary differences. However, there are new methodologies that are currently not used in forensic science, but used in other scientific disciplines that could be helpful for analysing both the inorganic and the organic components to help get at those exclusionary differences. So the aim of this study was to apply and assess the added value of some new quantitative methods to the analysis of soil for forensic soil comparisons. And so this was a multifaceted team that underdid this work. And so my laboratory at NC State looked at the organic components via DNA metabarcoding. The FBI lab did the traditional forensic exam, but also supplemented that with a new method of instrumental colorimetry. And then John Jay, and in consultation with Microtrace, looked at a new method for quantitating and characterising minerals by SEM-EDS. So this is the research team. Obviously, myself and Jack are here presenting today, but there was a large number of individuals who not only helped us with sample collection, but also helped us with processing the material in this. And so we just have a recognition of those individuals. And I want us to also point out that we also had the forensic examiners at the FBI as our subject matter experts in this project. So in order to test these three new quantitative methods, we needed samples. And so we collected surface soils from across North Carolina. North Carolina is not only advantageous due to close proximity to us, but it also represents diversity with regards to the parent material, which you can see here on the top map of North Carolina. Those different colours indicate different parent material. So it's quite diverse. It also has diversity with regards to elevation. We have a mountain region here, followed by a Piedmont area and a coastal plain. And within those differing areas of the state, we also have different compositions of and proportions of organic material to inorganic material. So it represents a wide diversity of the types of material that could be submitted in casework. So we sampled from 30 different locations across the state, which is this bottom, whoops, sorry, wrong clicker. Oh, now I'm going too far back. Oh gosh, okay, here we go. Sorry, the clicker's a bit delayed. So these are these different locations here in the mountains, the Piedmont, and the coast. And these were from NC State research stations that are used for teaching purposes that we got permission to sample at. So from each of these 30 locations, we had paired sites. And you'll hear us talking about this terminology of locations, paired sites, and subsite samples, so that's why we want to bring it up now. So at each of the 30 locations, we had a paired sites. So you can see here in this top image, these were about 100 to 300 metres apart. They had very similar parent material, but the land use differed between those A and B sites, as you can see here, and it's also given here in a graphic here. At each of the A and B paired sites, we also collected subsite samples. And so these were taken a metre apart from each other to check and assess that very small scale spatial variability. So we had 180 samples available for analysis with the new methods. So we, as I mentioned, we characterised these using existing methods and augmented them with the new methods. And we wanted to assess the utility of these new methods with three sort of main scientific questions. The first was, is it possible to separate those paired sites at a given location, A and B? The second question was, for those research stations where we collected more than one location, could we separate the locations? They're only kilometres to miles apart. And then the third would be looking at those locations within a region, coastal mountains and Piedmont. Could we separate those samples in those locations in the same region? And so for the interest of today and the time, we are going to focus on the utility of each of the three new methods, both in alone and in combination, for answering that first question on separation of paired sites. And I'm going to hand it over to Jack.
Okay, so in the interest of time, actually, I'll be covering the research that I did as well as the research that Libby Stern and her colleagues did. So they looked at these soil samples that Kelly just talked about using traditional methods, and they particularly looked at using polarized light microscopy to characterize and identify minerals that were in the soil samples. And they did this in a semi-quantitative manner in that they identified a particular element or mineral, and then they decided it was a major, minor, or trace level, depending upon its abundance. And then they characterized these, or they put them into a tablet, or oops, went back here. They put them into these tables right here showing the abundance. In addition, they also used a handheld colorimeter, and so they were able to actually measure the color of the soil samples in a quantitative way. And they were able to match or map that color into a quantitative space, the LAB color space. And the nice thing about that is that we can now use an objective way to compare two different samples based on their color. And we can provide a quantitative metric to quantify the difference in that color. And so what they'll use is the LAB values are then using this very complicated mathematical equation to calculate the delta E. And so, again, it's a quantitative difference between the color. And depending upon that magnitude of the delta E value shows you how far or how different those two colors are. So if we can see in this table, I'm going to go back here. Come on. There we go. Okay, so you can see in this table here, there are these different values for the delta E value. So low being a very close in color to a very large number being very different from one another. And just to give you some real world kind of rationale or understanding of these values, you can see these colors in these two samples here. So these are two sites. So this is site A and site B at location 56. And we can see that these two colors are actually quite similar to one another. Those are in the same site. And you can also see the delta E value is actually quite small, indicating that closeness of color. And we can look at site A and then also site B. But now if we compare site A to site B, those colors are quite different by eye, and that is captured in the value of the delta E. And so we can use this delta E value as essentially a threshold in which we can say that two colors or two soil samples look indistinguishable. So if we look at all of the sites, so we look at the subsites or in these samples we looked at, we can look at the within site variability of the range of colors, and we're trying to see can we exclude or not. And if you use the threshold of 10 in our delta E value here, we can see that when we look at just the bulk of the soil, we can only differentiate or exclude 5%. If we look at the fine fraction of the soil, we can only differentiate 3% of the soil samples. And if we then combine those two, looking at both the bulk and fine, we only can differentiate 7%, which is, indicates that the samples that are collected close to one another have very similar colors. Now, if we look at the sites, site A relative to site B, we can now look at these values down below here. And again, we're looking at our delta E, our threshold or cutoff. We can see now we can cut off or we can exclude 21% if we look at the bulk, 19 if we look at the fine, and if we combine both of those two in the same sample, we can actually exclude 28%. So that was a very, very cursory and very incomplete assessment of the work that Dr. Stern had done. Please, she'll go into much greater detail in a more complete picture in her presentation on Friday. Okay, so I'm going to talk about the SEM EDS portion of the research. And so this project first came about when I learned about this program called DTSA, which is Desktop Spectrum Analyzer. And among the many functions that it can do is it can actually model the EDS spectrum for a given elemental composition. So if I have, for instance, I've got the mineral titanite here, and we measured the elemental composition of that mineral, we determined this is the composition of it using a complementary technique. We can then take those values, put it into DTSA, and we can actually model what the EDS spectrum will look like, and that's what's shown here. We also then collected the EDS spectrum for that mineral, and that's overlaid on top of here. So clearly, you can see that the modeled spectrum and the empirical spectrum sit right on top of each other really, really well. So that gave us the idea of can we actually use DTSA to build up a database of minerals that we can then search unknown minerals for in forensic situations to see if we can identify the unknowns in the soil sample. So that's exactly what we did. So we searched the literature looking for published results of mineral compositions as well as introductory mineralogy textbooks. And we modeled all the spectra using DTSA. And so we have this large database of mineral compositions. We then took known minerals that we had from our reference collections where we knew that mineral. And then we essentially searched this database with these minerals to see if we actually could get to the right answer. And so the way we did that was we used the entire spectrum. We are not reducing the EDS spectrum to element weight percent values. We're using the entire spectrum, much like you search a FTIR spectrum in a reference database. It's the exact same thing we're doing here. And we did a bunch of several pre-processing, but we ultimately are using Manhattan distance as the comparison. So we have an unknown, we compare it to all the reference spectra using Manhattan distance. And the overall accuracy of the method was about 86% accuracy in the top hit, okay? The strengths were several different mineral classes, particularly opaques, but we also had some difficulties with minerals that experience or have what's called solid solution. That is where they have their mineral groups that have very large or wide range of elemental compositions, and depending on what specific composition will identify a particular mineral. So rather than trying to go down to the species level, we went and actually just bin things. We went to a more conservative characterization. So we're actually not identifying the mineral, we're classifying it into a group or a bin. So when we take now looking at the samples, the research samples, we collected or we did heavy mineral separation. We then back sieved those heavy minerals onto tape, embedded them in epoxy, ground them flat. So we have very nice flat surfaces, and then essentially treated the samples as if they were gunshot residue in a similar manner. And so with this method, we could actually characterize or classify 1 to 2,000 or 1 to 3,000 mineral grains per sample. So that gives us a lot of big numbers per sample to characterize. And so we use the abundance profile for each of those samples as the characterization of that sample, and then we did all pairwise comparisons using a metric called Bray-Curtis, which is used in the ecological research for comparing different sites based on living organism species levels. And so we're just adapting this to our method here with mineralogy. And so we looked at all of the sub-site or within-site scores, which are shown here for the Bray-Curtis values. And then these are all of the scores for things that are outside of the same site. Okay, so these are essentially would be the match, and these would be things that are known to be not match. And we also then plotted it using an ROC curve, and I show that our algorithm is actually doing quite a good job of separating out or getting the answer correct. And then interpretation of both of these data here allowed us to come up with a threshold, and that is a Bray-Curtis value that we're going to say, if you are at this threshold or below, you are considered indistinguishable or a quote a match, and if you're beyond this threshold, you are excluded. And so that value was 0.303. And then using that threshold, you can see our true positive rate is about 87%, and our false positive rate is about 16%, when we are only using SEM-EDS. When we combine SEM-EDS with the color, so we do first color as a first pass, then follow it up with the SEM-EDS, you can see our true positive rate drops down to about 80%, but our false positive rate drops down to about 9%.
Perfect, thank you. Okay, so Jack has talked about our two of our new quantitative methods. I'm going to touch on the third quantitative method, which is the analysis of the organic components using DNA metabarcoding. I'm just going to quickly walk through what the DNA metabarcoding workflow consists of. So for this, we had about 100 milligrams of soil that we subjected to a DNA extraction, where we extracted all of the DNA components that would be in that sample, regardless of its origin. We quantified how much DNA we had, we determined via the qubit, and then we amplified regions of the genome for each of these 4 taxa. So we're targeting arthropods, plants, bacteria, and fungi that can be useful for taxonomic identification, so DNA-based species identification. So we amplify those regions, we determine how much we amplified, and then we sequence using Illumina chemistry. And Illumina is the most widely used new sequencing method that many research labs and forensic labs may have. From that sequencer, we essentially get a sea of sequence reads, and so that's not very helpful at that time, so we have to do some processing steps. So the first thing we do is trim for quality. Then we run it through a software called Data2. This essentially will say, I have this sequence, and how many copies of it do I have in my sample? So we get a list of the unique sequences and their abundance in the sample. Then we take that list of unique sequences and search them against a reference database, similar to what Jack was talking about. You can think of it similar to a grocery store. You have a barcode, it looks it up, it tells you what that item is. For this purpose, we're searching sequence databases to tell us what that species is, or the taxonomic identity. So we do that, and then we get a list of the species per sample and the abundance in each sample. So then we have that data. How do we use it to do these comparisons? And I'm going to walk you through this slide. So the first thing I want to highlight is that very left panel. So we essentially get varying levels of granularity with this data. We can get down to the very sequence, which is what we have here is the ASV. So this is the very sequence. How many of that sequence do we have, and what is its taxonomic identity? That's very granular. And there's not been many studies that have looked at the impact of analysing data at different levels to see how well these questions can be asked. So we also looked at analysing the data at the family level. That's obviously less granular. You may be losing some information, but we didn't know. So we wanted to analyse our data at the sequence level and the family level. And then we also, these bottom two here, we wanted to exclude the top 25% of taxa, sequences or families, because those could be abundant in every sample and could be not providing actually any useful information for the discrimination. So we have those 4 different data levels, and then we also have the different taxonomic combinations here. We didn't know which would perform best under which circumstance and for what question, so we wanted to analyse them all. So 4 times 8, we have 32 different analyses per question, per location, per everything. So it's very complicated. We did a lot of statistical analyses, but today I'm going to talk a bit on the Bray-Curtis, what Jack just talked about. And I would just like to highlight, we just had this paper published as of Monday, so now this is Teague et al. In Diversity, if you're interested in finding more. So now I'm going to talk about how did the DNA analysis work when it was comparing that A to B. And so what we were able to determine was the biological communities that we recovered using DNA analysis did vary for those A and B at a given location, but it differed based on the target that was used and the level of the data. So you look at this table here, again, these are the 4 different data levels that I just explained, and these are the different taxonomic combinations. If there is 100 in that cell, that indicates that for 30 locations, that data set with that taxonomic combination was able to separate A and B. So again, we're looking for the highest number in these cells. So we can see that data with bacteria seems to work best for that A and B separation. And it all seems to work best if we have that very granular level, so that sequence level. And so we will be doing this for all these different comparisons and analyses. Notably, the samples in which we weren't able to get the A and B separation was for when we had mineral-rich samples, which makes sense. We are harnessing the organic component of these soils for the DNA analysis, so it would make sense that they're not separated as well with this approach. So again, I'm also giving a poster tomorrow, so if you want to know more about the DNA results, please come and see me tomorrow. And now we just want to touch on how well do these methods work in combination. Our end goal here is to try and identify circumstances and provide guidelines as to when each of these methods might be helpful, depending on the question that's asked of the examiner. So this is an example of a location where these new methods worked really well to provide differentiations between, again, our A and our B sites. So we can see here, this is location 61. These are the A and B. They look very different based on their land use. DNA worked well. Every data set of those 32 could separate the A and B. Our instrumental colorimetry had that value greater than 10, so that allowed for that separation. And this bar graph here is the mineral content analysed by SEM-EDS. So you can see these are minerals here along the x-axis. A and B are the different sites, so we can see there are differences in the mineral composition as defined by SEM-EDS. Notably, the mineralogy done by PLM only said that these samples, the A and B, were very similar. So we can see that the new methods in this particular instance are really providing better resolution. In full transparency, this is a location where the new methods didn't provide as much help as the traditional examination. So this is location 60. Again, the 2 sites here, they look quite similar in their land use, even though we tried to pick as much as we could separate. DNA only worked in about a third of those different data set combinations, so we would have to be selective as to which one we chose on a given situation. Instrumental colorimetry, also an SEM-EDS, you can see the profile looks quite similar, but the PLM mineralogy, the traditional approach, did identify one mineral that differed between the A and the B, so there was some discrimination there. So just to wrap up here, I know we've put a lot on you. So instrumental colorimetry is fast, it's non-destructive, and it gives that quantitative measure, which can be subjected to statistical analyses. The SEM-EDS is a very fast and effective way to screen and quantitate hundreds to thousands of minerals. It also can be transferred to instrumentation existing in labs, but it would need to be done by someone who has some expertise in mineralogy. The DNA is feasible from organic and inorganic material, again can be transitioned to a lab that's got molecular equipment and can permit that paired site, that A and B separation. We would love to continue this work on additional soil samples from outside North Carolina and then assess the impact of storage conditions on these results. Also, we really want to look at that small spatial scale variability and also more broadly within the regions. And with that, we would just like to acknowledge all the other people who have supported this work, NIJ for funding, and again, if you're looking for more information on either the mineralogy or DNA, please come see us. And I think we might have time for a short question.
We could take one question. Any questions? Also, I should note that those of you who are watching online can enter questions into the chat or Q&A. Hearing no questions, we will just keep to the agenda. If you,
Perfect, thank you.
You want to get in touch with Kelly and Jack afterwards, I'm sure they'd be happy to talk to you. Our next speaker is Dmitry Kurouski. He is Associate Professor at Texas A&M University. Dmitry.
Thank you for introducing me, and many thanks to NIJ for putting this event together. great location. A little bit chilly if you come from Texas, but I think it's not that bad. At Texas A&M, we focused on advancing the forensic analysis of hair. In our opinion, it's a very important task because hair is one of the most commonly found pieces of evidence at crime scenes. And obviously, that analysis of hair, forensic analysis of hair, can be used to establish a connection between a suspect and a crime scene or demonstrate the absence of such connections. Because the paramount importance of hair, obviously, people use light microscopy to examine its visual appearance and texture, and through this visual examination that is performed by a forensic expert, they can make a conclusion whether the hair has human origin or not, and if it's human origin, where it comes from, whether it's facial hair, scalp hair, etc. And to a large extent, although with many mistakes, the race can be predicted via the microscopic analysis of hair. The problem with microscopy is it not only requires a lot of training for the forensic expert to perform it right, but also it's very subjective, and as a result, there are mistakes made by forensic experts pretty frequently, and there was a study on this published in FBI report. As a result of the subjective nature of microscopy, other techniques should be utilized and deployed to advance the forensic analysis of hair. One of this technique is polymerase chain reaction, as well as its analog quantitative or qPCR. It's a great technique that can be used to identify the genes linked to the DNA, and it works not only if the hair has intact bulb, but also works if there is mitochondrial DNA present on hair. And by all means, it's the golden standard of forensics, but the problem is there are too many samples waiting in the line, and it takes months for the forensic laboratory to release results, and obviously all of this time the suspect will be in detention facilities, and it's not ideal. And I also want to mention that HPLC and HPLC-MS broadly used to look at illicit drugs and explosives that can be present on hair, and perhaps many of you that sit in this audience utilize these great techniques, again, to advance the forensic analysis of hair. In my opinion, what was badly missing is the fact that nearly half of people around the world dye their hair, and hair dyes are very specific in terms of their chemical composition, and if we find the technique that can be used to detect and identify dyes present on hair, pretty much read their chemistry, then we can dramatically advance the forensic analysis of hair, because through this type of evidence we can make a direct connection between a suspect and a crime scene, showing that exactly the same dye was used to color hair present on suspect and hair that is found in a crime scene. The problem is that when we look at hair, colored hair, we'll find that we deal with a very small amount of dye present on it, and the dye primarily covers only the surface of hair. So it's hard to find the technique that can be used to read out the information about hair dyes. Obviously, light microscopy becomes the technique of choice, and there are many light microscopy techniques, including Raman spectroscopy, infrared spectroscopy, circular dichroism, and fluorescence spectroscopy. The problem is that these techniques are not sensitive enough. Raman just doesn't see information, doesn't see dyes, the signature from dyes, and I'll show it to you in a second. Infrared spectroscopy primarily probes the bulk volume of the hair, and the signal therefore is dominated by vibrational signatures of keratin. And for fluorescent techniques, it's a great and nice methods. Just again, not enough dye signal contributes to the spectrum, and therefore we cannot have robust and reliable information about dyes present on hair. The technique that my group develops and deploys is known as surface-enhanced Raman spectroscopy, and this technique is based on the gold nanostructures that we add to the sample. As you can see here, we have a fragment of hair, and we can drop cast the solution of gold nanoparticles. And in this case, what happens, if light hits the nanoparticles, it will enable coherent installations of conductive electrons on their surfaces. These coherent installations of conductive electrons, known as localized surface plasma resonances, allow for a 1,000,000-fold enhancement of Raman signal Raman scattering that will come from the nanoparticles. And it's not only a great technique in terms of sensitivity, but also very good technique in terms of specificity, because what we get as a result of this type of analysis is the vibrational spectrum. And the vibrational spectrum is very rich in terms of bands that can be used to analyze and identify the dye present on here. Well, this is how it looks at the nanoscale. So we have hair. If we apply nanoparticles, we will have nanoparticles attached to the surface of hair. And it's great because dye is present on the surface of hair. So if we put nanoparticles where dye is, the nanoparticles will enhance and amplify Raman scattering from the dyes present on here. And by the way, if you are interested in doing this technique, please reach out to me. We'll be happy to share samples of gold nanoparticles so you can repeat and reproduce our results in your laboratories. We make nanoparticles, we make them very well, and our nanoparticles provide great surface-enhanced Raman spectra. So if you're interested, please let me know. And this is just an example of how this technique can be used to identify, to identify the colorant. So if you can see, if we dyed hair with iron sky blue, the semi-permanent colorant, we put nanoparticles on the surface of this colored hair, and we shine laser light on it, we get the red spectrum. And this red spectrum is very rich in vibrational signatures that originate directly from the dye. How do we know? We can get the reference spectrum of the dye itself, which is basic blue 77, and you can see a perfect match between the red and the black spectra, which tells us that although there are other components present in hair dyes, such as surfactants, Raman specifically see the dye itself. Now, if you look at the spectrum C is the signature of normal Raman spectroscopy. As you can see, normal Raman spectroscopy without the effect of dye amplification driven signal cannot reveal the information about the colorant itself. Finally, the hair without dyes in spectrum D does not have any vibrational signatures. And of course, someone can say, well, that's great, but how specific is Raman spectroscopy? To answer this question, my student Samantha Higgins acquired serospectra from over 30 dyes. And as you can see, vibrational signatures of these colorants on the left. And we not only look at them from perspective of presence or absence of vibrational bands, which is possible because if we turn the presence of vibrational band in black bar and absence of the vibrational bands in white bar, we can get a barcode that would be unique for every single colorant. But in my group, we employ chemometrics approaches, something similar like PLSDA or more advanced artificial neural networks to demonstrate the accuracy of identification of dyes present on hair. And on the right part of the screen, you can see a table with true prediction rate for every single colorant. And as you can see, when we built a model based on spectra that we acquired, and it was roughly 50 spectra acquired per class of dye, we get nearly 100% accuracy in every single case. The lowest was 76, and some of them were in 80s, which tells us that SIRS is very specific in terms of the dyes that it senses on hair, and one big model can be used to detect and identify dyes that are present on the market in the United States. What is also very interesting is that we can predict the manufacturer. And in our opinion, it's very important because every month manufacturers come with new colorant, and every year. And of course, if we don't have this dye in our library, the question is how can we enable its identification? And as you can see, still based on the spectroscopic library that we created, we can identify whether the dyes were manufactured by L'Oreal, Vela, etc. And not only this, but also we can predict these manufacturers with very high accuracies. And finally, I also want to emphasize that Raman spectroscopy, surface-enhanced Raman spectroscopy can be used for automatic prediction of the color that hair will possess, the dyed hair will possess. And again, this is possible by the use of vibrational signatures that we acquire in the lab and chemometric models that we develop. If you look at the left part of the screen, you will see average spectra that originate from different dyes belong to different colors, and on the right you can see chemometric model that predicts the accuracy of identification of these colorants. And you can say that, well, it's great. Indeed, we have a technique that is very sensitive and is very accurate in identification of hair dyes. The question is how easy this technique can be used to perform analysis on hair exposed to different environmental factors, because under normal circumstances, the forensic expert would deal with hair exposed to sunlight, to bacteria present in the soil, to water, etc. So in my group, we try to make this technique robust and reliable and useful for actual forensic crime investigations. And therefore, we have to model this environmental effect. And one of the environmental effects that we modeled was the UV radiation. And in this case, we dyed hair and exposed them to sun for roughly 3 months. And as you can see, that as time progresses, we see a decrease in the intensity of vibrational bands in the acquired spectra. And this is true for both permanent and semi-permanent colorants, which tells us that dyes fade. It's expected, and it's a very known thing, it's a very known phenomenon in art conservation sciences, where people deal with the fading of dyes in paintings. So exactly the same is happening here. As we can see, if we sample hair exposed to a very short amount of time, we can get signatures, but if more time passes by, then it's very hard to get exactly the same vibrational signature. Well, first thing that it tells us is we can use this to our advantage. We can monitor changes in intensity of vibrational bands to predict the fading rates. And it's very cool because, as you can see, the results of ANOVA, where every single dot is the mean and the bar is the standard deviation, for every single black and blue permanent and semi-permanent dyes, we can trace the duration of the hair exposure to UV radiation. And obviously, this was done in the state of Texas, and more work is needed to translate these results to environments in other states, including California and New York and in Baltimore, when it's much cooler and the UV indexes are not that great. And for every single day, the UV index was recorded, and if you're interested in this information, I'll be happy to share it with you. But also, as you can see, once the spectroscopic signatures are acquired, once we have the library, we can use this information to enable prediction of both colorants and weak with roughly 90% and above accuracy. These results tell us that with this spectroscopic signature in hand and chemometric models, we can enable highly accurate prediction of dyes present on hair, as well as duration of the dye exposure to the environment. Another environmental factor that we look at was hemolymphian water. Is the water that's present in the bottom of the lake. This water is enriched with microorganisms. And as we can see, the effect of these microorganisms is pretty strong on hair. In every single case, again, we use the same combination of black and blue permanent and semi-permanent dyes. We see a strong decrease in intensity of all peaks, which tells us that, again, we have a degradation, that microorganisms degrade colorants present on hair. And similar to the effect of UV radiation, we can trace the duration of the dye exposure, of the hair exposure to this environment. Again, as you can see, we have somewhat linear changes between the intensity of vibrational bands and the lengths of the hair exposure to the hypolymphium water. And again, if we build a chemometric model, we have pretty good accuracy of identification of dyes based on the spectroscopic signatures that we acquired. So as you can see, it's critically important to perform experiments not only in the lab under clean and nice environments, but challenge ourselves and get the information about hair exposed to various environmental factors. And I also have to mention that the effect of heat exposure on hair, and here we observe very strong changes that are triggered by hair exposure to heat at 220 degrees Celsius. That is present on permanent and semi-permanent colorants. And nevertheless, if we utilize chemometrics, we can enable highly accurate identification of colorants present on hair and exposed to the heat environments. Well, very recently, we challenged ourselves to answer the question, to what extent soil can alter the sears-based analysis of colorants. And to do this experiment, we dyed blonde hair with permanent and semi-permanent colorants, and we exposed them to three different kinds of soil. And what we found is very interesting results. We found that spectroscopic signatures of dyes change very, very quickly, almost after a week of exposure, which tells us that bacteria that are present in soils will degrade colorants that are present on hair. And once we do chemometric analysis, what we find is in some cases we can make this trace, we can predict the duration of the hair exposure to soils. As you can see here, for example, for permanent black and permanent blue, it works very well, but it doesn't work this way for other dyes. In some cases, we just cannot make this linear trace to predict the duration of the hair exposure to the soil environment. And what is also very important is chemometrics can help us for control hair and hair exposed to soil only for a week. Once we have more than 1 week exposure, we cannot build robust and reliable models that will enable highly accurate prediction of colorants present on hair. And it's not surprising because bacteria that are present in soil, they degrade colorants, and which results in very poor accuracy of surface-enhanced Raman spectroscopy. So as you can see here, we found a case in which sears is not great, but it's also very important to know for the forensic investigators that if hair spend more than 1 week in soil, then sears-based analysis of colorants present on hair may not be as robust and reliable as expected. Nevertheless, if we perform more bulk classification of the hair exposure in time intervals from 1 to 4 weeks and 5 to 8 weeks, then you can see for the dyes that we tested, we can predict the duration of exposure. So again, we cannot do it with 1 week precision, but for time blocks, it's actually possible, and it actually works pretty well. And in the very last part of this talk, I want to share our most recent result in which we investigated the effect of chlorinated and non-chlorinated water. What we tried to model in this case is the person submerged in pool, and pools are chlorinated, and investigate whether chlorine that is present in pools would change the sears accuracy of detection and identification of artificial colorants. And for this, we colored blonde hair with 4 different dyes, 2 permanent and 2 semi-permanent, and we submerged them into chlorinated and non-chlorinated water. So what we find is that chlorinated water, so non-chlorinated water by themselves, do not really change much sears accuracy doesn't really change much sears accuracy. As you can see, we have pretty robust and reliable spectra that are dominated by vibrational signature of the dye itself. But in this experiment, what we did, we put all 4 hair dyed by all 4 different colorants in 1 beaker. And as you can see, what we found is blue permanent dye leaked out of hair and contaminated other hair that were present in this beaker. And therefore, if this happens, we can have the leaked dye overwhelming the spectra acquired from hair that were dyed by another colorant. And as you can see here, it's exactly the case. So in case we have hair colored with a very strong dye, this signature, the signature of this dye can mask the vibrational signatures of other colorants present on hair. And this actually happened for 3 other colorants. And what we can say is, if this happens, we can predict the spectroscopic signature and the duration of the exposure of hair to water only for this dye. And for other dyes, unfortunately, it cannot be predicted. Nevertheless, we can predict the duration of exposure of both chlorinated and non-chlorinated dyes dye for chlorinated and non-chlorinated water. With all of this, I want to thank my group and Aidan and Isaac that perform the vast majority of work, NIJ for the financial support, and I have to say that we come to the end of the grant cycle. We published 10 papers in pretty good peer-reviewed journals that summarize the accuracy of series-based detection identification of dyes in the laboratory environment, as well as in hair exposed to different environmental conditions that I just described. We, in my opinion, put a very interesting review together with Aidan. So if you're interested, please read this paper. It summarizes not only series-based identification of dyes, but in general the use of surface-enhanced Raman spectroscopy in forensics. And it's also a very interesting fact that as a spin-off of this project, we had an undergrad that was very interested in forensic spectroscopy and forensic research, and the undergrad expanded this approach that we developed for hair to enable detection and identification of colorants in fabric. It's another very important area because, again, fabric, similar to hair, is often present at the crime scenes, and we deal typically with colored fabric. You can look at my clothes and your clothes, and of course the technique can work as great for detection and identification of colorants present in fabric as it works on hair. With this, I want to thank again, Greg, to you for inviting, and have to say that everything I presented does not reflect the NIH vision. It's the results reported by our lab. Thank you so much. I'll be happy to take questions.
All right, we do have time for any questions. Also online, please go ahead and submit any questions. If people in the room can raise your hand, I think someone will find will hand you a microphone.
Thank you. I really enjoyed this presentation. You mentioned that you manufacture the nanorods at your laboratory. Is that a difficult task to do? Could you explain it a little bit?
Yeah, it's a very good question. It's not a difficult task to do in terms of it's easy to make some nanostructures, but it's very difficult to make nanostructures that would give very good Search spectra. And in my lab, we spent the last year perfectioning our synthetic approaches to make nanostructures that enable really good reproducible signals and very strong enhancement factors that are necessary to perform this work. And like I mentioned, I'll be happy to share the synthetic procedures as well as share nanostructures to the respectful audience if you want to perform SIRS or any other type of measurements. We'll be happy to provide nanoparticles.
Dmitry, very interesting presentation. I'm just curious about the dye, the higher dye itself. Is complex mixture of different dye molecules, or it can be a mixture of different dyes, right? Do you have an idea of the composition of those higher dye?
Right. Products? Yeah, this is an excellent question. So it's different for different dyes. When we deal with semi-permanent colorants, we often deal with one dye. For example, basic blue 77 is the only dye that is present in this colorant. And in this case, it's easy. In the case of permanent dyes, we deal with phenyldiamines. These are small molecules that are not colored by themselves. And when developers pretty much oxidative agents, what happens is these phenyldiamines polymerized in the highly rich polymers with a lot of amine groups and azo groups and aromatic groups, and they're called Kurouski bases. Again, for this case, the nature of a dye is pretty much known. It's simple. But the problem is when we deal with semi-permanent dyes that have many different colorants in them. And to answer this question, what Aidan is doing in my group right now, he purchased every single component of most of colorants that we could buy, and we try to weight the Raman contribution of every single dye present in this colorant, because in many cases there are 5 or 6 or 7 dyes present in some semi-permanent colorants. And of course we want to know what is their weight in the search signatures that we get. Is it equal contributions, or maybe one dye gives 95% of signatures to the spectra we acquire. We do this work right now to answer this question. It's very important. Thank you.
Oh, actually, hello. You have covered quite a bit of interesting details. One of them caught my attention when you used chlorinated and non-chlorinated agitated water. So I have two issues just hit me. Is the chemical composition of two base water are the same? Is the only difference is the chlorination? And the other thing is, why did you use agitation? Is that like you try to steer the solution when you're doing it? That will add a lot of like mixing and turbulence and excessive like erosion of the coloring.
Correct. So water is exactly the same. We just added chlorine tablet to one of the beans, and it was not added to another one. And you're completely correct. We try to mimic the actual pool environment in which water is circulated. Therefore, we use agitation. But right now, we work the body farm in San Marcos, and we will submerge a body with dyed hair in the pool, and this pool will not be as heavily agitated. We will put the pump to mix water, but we do it to mimic the environment that the body can experience in the pool where water is circulated.
All right, please join me in thanking Dmitry one more time.
Thank you.
Speaker is in fire research, so NIJ has supported fire investigation research for years. Next speaker is Matthew DiDomizio. He's a research engineer at the Fire Safety Research Institute at Underwriters Laboratories.
Good morning, everyone. Today I'm going to speak to you about a study we did at the Fire Safety Research Institute in collaboration with the Bureau of Alcohol, Tobacco, and Firearms, and of course funded by the National Institute of Justice, looking at heat transfer and fire damage patterns on fire-exposed walls. We'll get into the fire model validation part of that as well. I think this is not... There we go. So this was a 2-year research study and a collaboration between ATF and FSRI, as I mentioned. And the objectives of this study were really twofold. The first was to develop validation data, specifically for fire models, that addresses 3 validation spaces. One of those being the thermal exposure to walls in a fire. And that would predominantly be characterized as a heat flux, an incident radiative and convective heat flux. Heat transfer within fire-exposed walls, so that would be heat transfer through those walls and the consequential decomposition of them, as well as fire damage patterns arising on fire-exposed walls. And we'll dig into that a lot in this presentation. The secondary objective was also to advance our ability to predict fire damage patterns on walls using fire models. We conducted a total of 63 experiments in this study, and culminating in releasing publicly available data set. So that data set is available for the entire research community, both fire investigators as well as fire model practitioners and fire researchers, to validate and expand the capabilities of fire dynamics models. In addition, we have produced novel findings for the investigation community, and we'll be getting into some of those today. So a little bit of an introduction on heat transfer in fire environments. A fire will develop over time from some initiating event, whether that's a piece of furniture, a liquid fuel spill, a single origin, and from that grow to involve multiple combustible sources, potentially an entire structure. As that fire grows over time, its size can be characterized either by a rate of heat release, that is sort of a measure of the rate of change of energy content within that room, or gas temperature, a measure of how much, again, thermal energy is contained within that room. What can happen is there can be a transitional event, which we call flashover, which is a transition from single items burning to all items in a compartment burning. And that's indicated by the pictures here. And you can see sort of when there is a pre-flashover environment, the interaction between the fire and the walls of the compartment are predominantly driven by that heat exchange between the flame and the wall. However, in a post-flashover environment, the entire gas environment is very high temperatures and you're transferring heat by radiation and convection to the walls, resulting in very different damage patterns. Reason being, fire damage patterns are predominantly related to the thermal energy transfer from the fire. What's shown in the bottom figure is sort of the net energy transfer that we would consider when we're looking at the decomposition of the wall and consequently the fire damage patterns that will form on it, where we consider both the heat transfer to the fire exposed side and the heat transfer from the unexposed side. Computational fire dynamics models can be used to predict a lot of things that can result from a fire and that are of relevance to fire safety engineers and fire researchers and, of course, fire investigators. Relevant to this study include predictions of gas temperature and velocities within a fire environment, heat transfer occurring on, to, and through objects, solid objects, for example, walls or other combustibles, the temperature of those solid objects, and then the mass loss occurring of those combustible objects. And mass loss is something that would be predicted by a fire model by some defined kinetic mechanism. So the required inputs to that model, generally something about the geometry, something about the fire or the fuel characteristics, and that would include both the origin as well as the entire compartment, and then information about the scenario, how the fire will evolve over time. So how a fire model can be used by an investigator then would first, step one, develop a hypothesis, define the scenarios of that hypothesis based on factual evidence, and then aim to reject those hypotheses by comparing predictions of the fire model to physical evidence collected on scene. When a hypothesis has been rejected, then of course develop alternatives until you can develop a credible hypothesis. That gets us into fire damage patterns. So fire damage patterns are physical evidence that are present in post-fire environments. It's not something that can be tracked over time. Obviously, it's something that we have a cumulative measurement of at the end of the whatever the situation was. They are they're characterized rather by identifiable shapes, and those shapes can be demarked by either lines or areas or regions. But we can identify lines of demarcation, for example, in this photo, which shows a piece of drywall that's been exposed to a gasoline fire. And you can see the fire-affected region has been charred, and paper has been sort of bubbled up and exhibits this alligator skin-like appearance. But then surrounding that is a region of discolored paper, and then eventually transitioning to the virgin paper. Regarding pre-flashover / post-flashover, this is what I was alluding to before, where when there's a pre-flashover fire, we can expect fire damage patterns that are related to that source fire, which is actually what's indicated in the photo. So that brief background sort of defines the motivation for this study, where there's a motivation specific to the fire investigator, being a fire model can be used to predict temperatures, it can be used to predict heat flux, and it can be used to predict mass loss occurring on walls in a fire scenario. A fire investigator can come in after the fact and identify the final state of some sort of damage scenario, with that damage being directly related to some heat exposure. And then the missing link is the mechanism that ties that thermal damage or that thermal exposure to the fire damage. And then secondarily, we have a motivation from a fire model validation perspective, which is to develop a comprehensive and useful data set. The scope of the present study was limited to pre-flashover fires. Post-flashover fires also of interest, in particular because a post-flashover fire can obfuscate patterns that may form pre-flashover. And also the current study was limited to gypsum wallboard in terms of materials on which fire damage patterns would form. So all the experiments were conducted at the ATF Fire Research Lab facility in Beltsville, Maryland. Experiments consisted of a freestanding wall placed adjacent to a fire source, all located under a calorimetry hood. It's calorimetry, not colorimetry. So this hood is designed to essentially measure the rate of heat evolution from the fire and any other combustible objects, i.e. the wall. The image here shows a top view where you can see a wall located adjacent to the fire. And just pointing out those dimensions, each fire had a characteristic size, so a base area, as well as a spacing from the wall. And that spacing was designed so that we would avoid direct flame impingement from the fire to the wall. But if the wall ignited and flames became seated on it, so be it. But the idea here was to get intense fire exposure, but not necessarily direct flame impingement, which would be a different kind of scenario. The types of fuels considered were a gas burner. That gas burner was characterized to be a 500 kilowatt burner. That half megawatt was based on the size of the hood that we were testing under, a 1 megawatt hood, but also important because everything was based on the size of the available materials, and the size of a most common size, rather, of gypsum wallboard would have been 4 feet by 8 feet. So if we want to get a much larger fire than that, we would need larger drywall. Larger drywall, we would have to create joints and mud those joints, and we wanted a pristine, continuous material for these experiments. Other fuels included wood cribs, a wood crib with a polyurethane foam slab, which is sort of a furniture surrogate, an upholstered chair, and a couple of different liquid fuel fires, gasoline and heptane. Here's a photo of the experimental configuration. On the right side shows the wall under the calorimeter hood. The wall is supported by steel frames, and the image on the other side shows, I believe that's a gasoline fire. And so you can see the flame is not directly impinging on the wall, but the wall is experiencing fire exposure. This sequence of images here shows a wood crib fire as well as a gas burner fire. Once again, pointing out no direct flame impingement. I believe one of these is a movie. Yeah, there we go. So you can see the general scale of flame height relative to wall height, where the tops of the flames are reaching the top of the wall, but the region of most severe radiation, most severe thermal impact, is sort of in the mid-region of the wall. This sequence of images shows transition for a gas burner fire from just starting to discolor the gypsum wallboard. So you can maybe sort of make out in these images a little bit of discoloration circled in that green line. Transitioning to the middle image, which shows that the paper is starting to char and discolor. Transitioning to the final image where you've actually got paper being burned off and the alligatoring appearance is just beginning. And here's another sequence of images which shows wood crib fire. And you can see much larger fire damage pattern that's being formed, in particular near the base of that fire, not so much near the top of the fire. Fire exposures, I mentioned previously, are characterized by heat flux. And one method for measuring that heat flux is using a gauge, an instrument called a heat flux gauge. A Schmidt-Bolter heat flux gauge is really popular in fire science, and that's shown there, the copper gauge. Obviously, the problem with such gauges is that they can only be used to measure heat flux at a single location. So that gauge in particular is a 1-inch diameter gauge. You can imagine spreading those out over a 4 by 8 foot wall. You're either going to need a lot of gauges to characterize the variation in heat flux over that wall, or you're going to have a very coarse, granular understanding of that heat flux. So alternatively, One method that's available is an extension to a plate calorimeter method or a plate thermometer method, where we take a very thin piece of steel, paint both sides of it in a high temperature, high emissivity paint, characterize the emissivity of that surface, and direct an infrared camera at the unexposed side. In that manner, the plate is exposed to the fire, the infrared camera measures the temperature rise, and from that we can deduce heat flux over the entire surface of the wall. So this is a method that I and my colleagues have been developing over the years, and as a result of this NIJ project, we've formalized the methodology as well as developed open source software for the methodology, and we've called that HFITS, heat flux using infrared thermography over planar surfaces. So from that QR code, or if you just Google HFITS, you can find both the papers that we've written as well as access the Python code to actually run the software. And so I'll show you some examples of the application of HFITS. So here you can see we've got a heptane fuel located directly in front of the wall, and I've got an infrared image sequence as on the bottom, and then in the plot on the right shows the derived incident heat flux to that wall. So as the fire grows over time, you can see that the region of greatest thermal impact is in the center, as we would expect, center, left, and right, and more towards the bottom. But you can also see as the drywall ignites in the infrared image, as the paper on the drywall ignites, you're actually going to see a ring of heat sort of spread out from that origin, and that is the paper catching fire and heat transfer directing through the back of the wall. Again, that is an infrared measurement on the back side of the wall. So it's a really powerful method that lets us characterize heat flux over the entire surface. Now we know what every single location on that wall, we know what heat flux it was experiencing, and after the fact, we know how much mass loss is occurring and what the visual damage patterns were. So now at every single location, we have mass loss, heat flux, and damage patterns. The damage patterns were just measured using photography. A regular grid was added to the wall just using a chalk snap, and image segmentation was used after the fact in post-analysis to identify where the areas of demarcation were, separating charred from uncharred region, as well as lines of demarcation around that edge. The mass loss was simply measured after each experiment. The drywall was removed, placed on a table, cut into regular 8-inch squares according to that grid, and then the mass and dimensions of each of those pieces of drywall were measured. And from that, we could assemble a contour, understanding the mass loss over the entire surface. So looking at the results from this study, this first plot here shows the heat release rate of all the fires. Again, I mentioned 7 different fire types. For each fire type, there were 9 experiments. So each of these heat release rate curves shows the energy release by the fire averaged over 9 experiments, and that's the dark line, and then the shaded region is the min and max. This just lets us show really the range that we got. And one of the good findings here is that overall the fires were quite repeatable. So we know from these measurements that if the fire size is the same, then the heat exposure to the wall should be the same. And we're demonstrating that for the gas burner, for example. You can see a nice steady heat release rate and very minimal change between experiments. So this image here on your left shows, it might be a bit hard to make out, but there are numbers on the image of the wall. And each of those numbers corresponds to a heat flux gauge. We did do some experiments with gauges installed in the walls in addition to the planar heat flux measurements. So those gauges, location 1, 2, 3, 4, and 5, 1, 2, 3 down the middle. The plots show heat flux measured over time. And so this is what is characterizing our thermal exposure. This is what we expect to be related to damage patterns. And you can see in the images at gauge 1, 4, and 5, we have a relatively low heat flux. And at gauge 2 and 3, we have a relatively high heat flux. And referring to the image, you can see gauges 2 and 3 are located in the most fire-affected region, the most fire-damaged region. And so clearly, those regions have experienced more heat flux and experienced more damage. So we have that one-to-one relationship. This image here, the sequence of images, shows a fire damage pattern for a heptane pool fire and 2 contour plots. One of those contour plots is the mass loss that was measured over the entire wall. Again, the contour being assembled from all those small little 8-inch by 8-inch samples, as well as the heat flux measured over the entire wall using the HFITS method. And so clearly we can again see that the most fire affected region, the most fire damaged region, is consistent with that region which experienced the greatest heat flux and the greatest mass loss of the drywall. So this image here shows the gas burner, one of the gas burner experiments, and here is a direct comparison between heat flux and the observed damage patterns. And what you can see is that the line of demarcation corresponding to the transition between the charred and virgin regions of the drywall aligns with what appears to be a dark region in the contour of the heat flux to the wall. So does this actually have any relevance? Does a value of cumulative heat flux over the entire test actually correspond to some level of damage, characteristic of a transition between a charred and an uncharred region? Maybe. Certainly in this test, it appears to be much darker in that region. And by comparing different exposures, different fire types, different wall types, maybe we can identify some trends. Maybe we can identify some reasons for that. The other contour, so one of the contours shows heat flux, the other one shows the gradient of heat flux. And that is simply taking the maximum of the X and Y gradients of that value. The gradient shows perhaps some indication of the edges of that line of demarcation. The gradient is of relevance potentially to investigators, being that NFPA 921, the Guide to Fire Investigation, defines a damage pattern or an area of demarcation as a transition in the gradient of the magnitude of a fire effect. And so we looked at the gradients as well. So that was heat flux. This is mass loss. So here again, we can see very similar patterns as with heat flux, where the mass loss that occurred on the wall tended to be much greater in the region of demarcation indicative of char, the fire damage. Again, whether those values correspond to anything meaningful, we'll look at in a moment. Also shown, gradient. The gradient of mass loss appeared to be a little bit more representative than the gradient of heat flux, but again, not perfect. We looked at it. Finally, here is a summary of those damage metrics. So over every single fire type, and I've only got 5 fire types of the 7 shown, because the other 2 were more for heat transfer validation stuff. We really only looked at these 5 for damage patterns. One of the things that we identified was that the damage metrics, the values of those metrics corresponding with the lines of demarcation, tended to be consistent across all 5 fire types. In particular, the mass loss. So in other words, the mass loss occurring on that line of demarcation separating virgin and char tended to be consistent across all the different fire types. And it tended to be consistent at a very particular value. So how can we use that piece of information? Well, we now have established a direct link between fire damage patterns and fire exposure. And that key link is not the exposure itself, but the mass loss occurring in the wall. How do we predict mass loss? Using a fire model. The fire model predicts the exposure. The exposure then dictates the mass loss. And so now a fire investigator has a direct pathway to predict the occurrence of fire damage patterns, and via this test hypotheses and hopefully improve the utility of fire models as a tool to investigators. All right, that's it. Thank you all for listening, and please let me know if you have any questions. Okay.
Unfortunately, we don't have time for questions. I encourage you to get in touch with Matt afterwards. Our next speaker, last speaker of the morning session, is David Stoney from Stoney Forensics. He's going to talk about a project on non-identifiable finger marks on ammunition. David.
Thank you, Greg. So I want to talk about, my monitor's still got the last talk on it. Not sure if that can be changed, but it would be useful. Is that, so the monitor up here has the prior talk on it?
It's going to be delayed, so if you could use the side monitors for now, that would be great.
Okay.
Thank you. Sorry.
So I'm going to talk about non-identifiable finger marks on unfired ammunition in handguns. Of course, it's an NIJ project. We thank you very much for the support. This work was done with a number of collaboratives. Pat Wertheim did the finger mark screening and annotation. Measurements of associative value were made with the cooperation of the University of Lausanne. Statistical data analysis was aided by Muida Cuellar of the CSAFE and University of Pennsylvania. And subject matter experts assisted us in our research. I'm going to talk about these things, non-identifiable finger marks in general, the specific project goals, research questions, and design, what we did, the results and findings, spend some time on discussion points, and then talk about next steps. Well, a non-identifiable finger mark. Non-identifiable finger mark is a finger mark with insufficient characteristics for traditional identification. It nonetheless has sufficient details for lesser degrees of similarity and a probability of occurrence within a population. It's very important to stress that this is not meant in the traditional way that finger marks are used. It's not a confirming identification, and it's a fundamentally different form of evidence. Currently, we use only the clearest, largest finger marks. These are good prints that could be identified or could be used to exclude somebody. But prints that don't meet that threshold, we set aside, and we typically don't look at them at all, and they're not used. There's a changing perspective regarding finger marks. The traditional perspective is as we get more detail in the finger marks going upward on this chart, the expert makes a judgment of the sufficiency of use for either identification or exclusion. There are expert differences in that decision, but it's an all or nothing affair. The perspective that we're moving towards is one more like on the bottom. We again have more detail in the finger mark going up on the scale on the right, on the left. But the pyramid is indicating now different degrees of selectivity. And what we look forward to is a time when we can measure how much selectivity there is in the detail within any given mark. Of course, we've rejected the model that's in the back. We don't allow for a it's him, it's not him kind of decision. But as a consequence of that, we're moving towards this other point of view. And that's a point of view that doesn't reject the use of non-identifiable finger marks, but would look towards them for what degree of selectivity do they really have. Our prior research has shown several things. First of all, the non-identifiable finger marks have high utility. They have high associative value. They're readily collected and they're found in important places. So they're useful. Also that they have frequent occurrence. And they're found very often and in high numbers. So the prior work's been published, but it's shown those main two effects. They exist and they have potential usefulness. In this project, we wanted to evaluate the occurrence and associative value of non-identifiable finger marks in a specific location that's on unfired ammunition that's been loaded into handguns. why that particular use? Identifiable finger marks are not found very frequently on handgun ammunition that's been loaded into firearms. It's an area that's been extensively studied. People have gone over and over trying to find methods that will more efficiently look for these finger marks, that will find them more often. And if you look through literature, you'll find 2 or 3 a year of studies that have looked at this particular problem. It's an area that is litigated very often. So the penalties that there are, if your client, from a defense attorney's perspective, if your client has use of a handgun in a crime, they're facing a significantly different penalty than if they didn't. If you're a felon and you're found in possession of a handgun, you can get your promotion revoked. You can get much higher, stiffer penalties. So it's an area that is very much of concern to the courts. So it's an area that we seldom find identifiable prints, and it's an area that is extremely important in litigation. So a research question, well, how often do non-identifiable finger marks occur on naturally loaded ammunition, and what range of associative values could be expected? If we find that these occur pretty often and that there's high associative values, then it's important that we move to change the paradigm from throwing out the ones that don't meet a threshold of identifiable. On the other hand, if we don't find them very often, then let's move on from it. Let's stop spending research money and effort to look at this problem again and again. So our research design, we collected unfired ammunition from a reasonably representative set of handguns. Then we applied methods that are well established to visualize and assess the friction ridge detail that's found. We measured the expected associative value with a well-established method to determine how rarely these finger marks occur, and then measured the rates associated with different types of ammunition and different types of handguns. Reasonably representative ammunition. We took volunteer handgun owners loading their own handguns. We separated into 2 different handgun types, semi-automatics and revolvers, and we developed 4 different cartridge size classes. Prior work has looked at the different sizes of ammunition for identifiable prints. We made some groupings of the ammunition, so when I talk about 38, it includes a 38 special, 357, 9mm, and so forth. The visualization method that we used is to take a sequence of photographs, rotating the bullet around, and taking the pictures of the cartridge from different sides, and then stitching them together and making a kind of a panoramic view of the cartridge case. This is a published method going back to 2015. There are more modern methods to do this type of photography, but they're expensive. Our criteria for the finger marks, any marks with 4 or more minutiae in the context of the ridge flow, were retained in the study. In prior work that's been done on ammunition, if an expert didn't judge it as identifiable, it wouldn't have been recorded. Our measurement of associative value used a method that we've published on before. It's based on expected score-based likelihood ratios. A score-based likelihood ratio that we use is based on PhD work at the University of Lausanne. It was been modified by a distortion model that I'll talk about, and then it's been calibrated within this database. So the method is by no means one that is universally accepted, but it's one that's well understood and has been around for quite a while. What does it do? Score-based likelihood models contrast two scores that are generated in a well-defined system. So the green distribution represented on the left is a background score. If we take this finger mark, compare it against a million other marks, and you get a score, and that's that distribution. The red distribution on the right is comparing the mark and getting a score with that mark against a distorted version of itself. So we have a distortion model that takes a finger mark, distorts it in all the ways you'd expect from different movements of putting that mark down, and then checks the score versus itself. The likelihood ratio comes from comparing the probability that you get a particular score in the one distribution, the green one, versus the other, the red one. And it gives you a ratio of those two probabilities. We looked at 1,263 rounds of ammunition from 164 handguns. What we have is a series of different caliber classes, revolver versus semi-automatic, and the different classes, as I mentioned before. So of these, on these rounds of ammunition, we found 479 finger marks with 4 or more minutiae on 345 rounds of ammunition. So it's 27.3%. The percentage of rounds across the different categories range from 20.4 to 44.9. So a low 22 revolvers, which is kind of expected for the smaller cartridge size. But they range from 20.4 to 44.9. So they're present. Now this is taking marks with as few as 4 points of identification on them. But this is contrasted with, in that same set, of those 479 marks, 64 of them were judged to be identifiable. And those range from 1% in the 22 automatics to about 14% in the 38 revolvers. Now, this is consistent with prior work that's found like, hey, wait, basically 1 to 5% of rounds of ammunition have a identifiable finger mark, so it's not worth it for my lab to do it. But if you extend your consideration to look at these non-identifiable finger marks, you get nearly 6 times as many rounds of ammunition that will show some means to associate an individual. Well, how many minutiae are we really finding in these marks? For these 479 marks, we've got a breakdown by the different types of guns, revolvers, semi-automatic, and the different sizes of the ammunition, and the number of minutiae across here, going to larger amounts on the right. So 200 of the marks with 4, and 113 with 5, and so forth. Now, these are all with different numbers of rounds of ammunition in each of the categories. It's a better view of it if we look at, for each handgun and ammunition type, what percentage of rounds will give us a result with how many points of identification in it. So that's what this chart is. Along the vertical scale, we've got the number of finger marks per 100 rounds that are showing a given number of minutiae. So for example, for the 32 semi-automatics here, we've got a range of this many finger marks showed 4, this many showed 5, this many showed 6, and so forth as we go along here. So we're getting percentages of marks. If you I'm reading that along the top there. 57% have 4, 32% have 5, 24 have 6, and so forth. So 4, 5, and 6, those would be not considered identifiable in almost any laboratory, and those marks would be set aside. For the different categories of ammunition and handgun type, they break into these 3 general groups. The 35 semi-autos that I talked about are in the middle. 45 and 38 revolvers have pretty higher concentrations across the board, and the rest of them group along the bottom. Well, what does it mean in terms of probability of occurrence or associative value? There's a fairly direct relationship between these expected score-based likelihood ratios and the number of minutiae that you get when an expert codes it. And what's on the vertical axis here is the logarithm of the ESLR rating. That's, again, the ratio of probabilities of these scores, and the number of minutiae along the bottom. What does that translate to? These are associative values that would be indicated by a given likelihood ratio. So 4 minutiae, approximate ESLR, and approximate associative value, 1900. So those are the values that we'd expect. Here we've got 5 minutiae. We have an approximate ESLR for that of 20,000, which means that the amount of associative value we have is about the same as an event that would occur 1 in 20,000 times. It's a significantly high associative value. And at 1900, I could say, okay, well, maybe that's not worth going after. But if we have the opportunity to get a type of associative evidence that is equivalent to finding something 1 in 20,000 times, why aren't we doing it? So here's at least the way I'm using the language here so that it's at least transparent. An expected score-based likelihood ratio of 1 in 20,000. What does it mean? Well, there's the long version of it. The probability of the score under the prosecutor's hypothesis, the mark is from the defendant's finger, is 20,000 times the probability of the score to the defense hypothesis. The mark is from a randomly selected finger, not the defendant's. Okay, so that's what we're measuring. A likelihood ratio of the same magnitude would result from a characteristic that occurred randomly once in every 20,000 times. And so in my slides, and as I'm talking here, I'm saying, well, the minutiae configuration of the mark is rare, 1 in 20,000. But that's a leap, and it's an explanation of how much strength is there in this associative value. We don't have statistical methods that will calculate the bottom line, but we have statistical methods that will do the top 2 paragraphs. So summary of the project findings then. If we look by round of ammunition or by cartridge, this is what we get. The percentage of rounds with marks rarer than 1 in whatever. And again, this 1 in 20,000 number that I'm kicking around, 17%, 18% of rounds will show a mark at least that rare or more rare. But that's for individual rounds of ammunition. And of course, in a case, if we're talking about marks on a loaded handgun, then we're interested in the handgun load. So we have a number of shots at getting a good mark. So here we have, going again with that 1 in 20,000 figure, 52%. So in our study, naturally loaded handgun ammunition, 52% of the loads had a mark that would be as much value as an event occurring 1 in 20,000 times. So the project findings have a clear demonstration that you do have a common occurrence of non-identifiable finger marks of high associative value on loaded handgun ammunition. We found differences among handgun types and sizes, but in all of the classes, even on 22 caliber rounds, we regularly find rounds of ammunition that are of high associative value for their finger marks. Revolver ammunition in the 38 and 45 caliber classes shows about 4 times as abundant marks as do the other types. So some discussion points. Well, the methods and approach were designed to meet the research goals of how often do these occur on naturally loaded ammunition and the range of associative values. It was collected from a reasonably representative set of handguns. We used well-established fingerprint visualization methods, and the associative values are measured using one method that's at least well-defined and reproducible. But there are a number of things that would sort of limit the findings or are worth stressing. During ammunition loading and collection, we didn't assess factors that are known to affect finger mark deposition. And they were intentionally left uncontrolled in this study because we're trying to get a representative range. But the loader's susceptibility to leave finger marks is known to vary. The cleanliness of the hands, the presence of corrosion on the surface, and the technique that the user uses to load the handgun. So these are all things we did not specifically control. The effects could be investigated if we'd like to, but the contribution to these would be limited as they're usually unknown in a case. You can't go back and check most of these things. We could have looked at different classifications of the handguns and the ammunition, and it might help with policy decisions, but it wouldn't change the overall results. Likewise, with the fingerprint visualization methods that we used, we did not use those on all the cartridges. We stopped if we found a finger mark meeting our criteria with the cyanoacrylate. And we also ignored marks that occurred on the projectile surfaces. So each of these things could, in fact, change the results, but they're unlikely to go in a direction of saying there's less. I can conclude at that point. Thank you.
We have time for a question or two. Question online, go ahead.
Yeah, am I on? This is a multi-part question. Does the ESLR take into account the commonness/rarity of minutia types, or does a minutia just count as one no matter? Or with such limited friction ridge detail, does it not matter?
I'll start with the last one. It very much matters. Secondly, what I presented in most of the slides here were numbers of minutia. But in fact, the ESLR, the expected score-based likelihood ratio, responds very much to the point that is inherent in the question. Different configurations of 4 or different, so 5 or different whatever, will show a range of expected associative values. And that is taken into account by that ESLR measurement. So that's the motivation for using it, in fact, rather than relying upon expert scoring and counting of minutiae.
Hi, David. I got a question for you regarding these numbers. How to present them to the jury? So what is the message that you need to convey to the jury? And how we have to train our people to go to testify to these numbers, if any?
No, it's an excellent question. It's unaddressed by any of my work, but it's also not. It's not a question that's specific or only in the context of this particular way. We have to learn to do that. If we're not going to rely upon experts and say, tell us the answer, and we're going to in fact go to experts and say, do your best to give me the scientific basis for what would be your opinion, then we have to have a way that the courts will accept us to answer that question. So it's a problem. It's an unresolved problem. I have my way that I would do it, which is pretty close to the sequence that I put up there. But it's any expression of a numerical summary of associative value is going to have that difficulty with it. And we all need to engage in that, but I don't have an answer to that.
All right, please join me in thanking David one more time.